Welcome to today's Explainer. We're diving straight into a high-density, PhD-level review of Med 144 endocrinology. Think of this as a lossless, uncompromising look at hormone taxonomy and receptor mechanics, brought straight from the university lecture hall right to you. We're literally not leaving a single biochemical pathway or clinical example behind here. This module is famous, or maybe infamous, for its rigorous exam relevance, so, get ready for a highly structured, deep dive breakdown. Okay, let's dive into this roadmap. We'll be moving through 6 precise phases, starting with scope and structural classification, then amino acid-derived hormones, followed by vitamins, peptides, and steroids. After that, we'll shift down to mechanisms with Group 1 intracellular signaling, PPARs, and Orphan receptors, and finally, Group 2 membrane transduction. All right, Section 1, Scope and Structural Classification. Let's define our taxonomic bounds. So before we can actually categorize anything, we have to define our parameters. This specific endocrinology module is strictly centered on 5 core chapters: the pituitary, the adrenal glands, diabetes, the thyroid, and bone and mineral metabolism. What's fundamentally excluded here? Reproductive endocrinology. We will not be covering pediatric, gynecologic, or transgender-related endocrinology today. Within those strict bounds, our primary taxonomy divides hormones into 4 major structural classes. We've got our amino acid derivatives, vitamin derivatives, peptide hormones, and steroid hormones. Memorizing this overarching architecture is absolutely crucial. It's incredibly high yield for exams. Honestly, mastering this basic breakdown regularly yields maybe even 2 questions right off the bat. Section 2, amino acid-derived hormones: tyrosine, tryptophan, and systemic output. Let's bring some energy to our first structural class and rigorously unpack these derivations, because everything here originates from just 2 amino acids. First up, tyrosine. It gives us our thyroid hormones, T3 and T4, plus the catecholamines secreted by the adrenal medulla, adrenaline, noradrenaline, and dopamine. And this brings us to the exact 80-20 adrenal rule. 80% of the adrenal gland is the cortex and 20% is the medulla. But of that 20% medullary output, exactly 80% of it is adrenaline. Next is tryptophan, which yields serotonin and melatonin. Now here's a fascinating fact. Serotonin's most abundant source isn't actually your brain. It's the enterochromatin cells of your gastrointestinal tract. Serotonin drives alertness and activity. Clinically, low levels are directly linked to depression. Melatonin, conversely, comes from the pineal gland. Finally, definitely commit the dose hormones of happiness to memory: D for dopamine, O for oxytocin, S for serotonin, and E for endorphins. And this brilliantly illustrates how the interaction between these 2 hormones dictates the exact chronobiology of our sleep. During the day, light hits your retinal ganglion cells, sending signals to the brainstem's Raphe nuclei to pump out serotonin. As darkness falls, usually around 9:30 p.m. to 10 a.m., that reduced light exposure is supposed to trigger the conversion of serotonin into melatonin. But here's the critical clinical reality. Gadget and phone exposure completely derails this process. If you're staring at a bright screen until 1 a.m., you are forcefully maintaining serotonin stimulation. You drastically delay the serotonin cutoff, which means you're going to completely miss the critical 2:30 a.m. melatonin peak required for truly restorative sleep. Section 3, vitamins, peptides, and steroids, molecular weights, and zonal origins. All right, we're shifting gears to a high-density data compression. I really need you to evaluate these structural boundaries like an examiner would. Small peptides have fewer than 50 amino acids. This includes all your hypothalamic hormones, ADH, oxytocin, and uniquely ACTH, which is the only small peptide from the anterior pituitary. Large peptides exceed 50 amino acids. You have to know the exact numbers here. Growth hormone has 191 amino acids. Prolactin has 199. Insulin has 51. PTH and renin also fall into this class. Finally, we have the glycoproteins. These are massive, over 200 amino acids, and they share a common alpha subunit. Examples include TSH, FSH, and LH. But here's a vital biochemical distinction. Glycoproteins are mostly protein with a small carbohydrate subunit. Do not confuse them with glycosaminoglycans, which are exactly the opposite: high carbohydrate with very little protein. Now let's map out the steroid hormones anatomically, moving from the outside in. The outermost layer of the adrenal cortex, the zona glomerulosa, gives us aldosterone. Moving deeper, the zona fasciculata produces cortisol, and the innermost layer, the zona reticularis, is responsible for the adrenal androgens, specifically androstenedione and DHES. I want to highly emphasize DHES, dihydroepiandrosterone sulfate, because it is absolutely the most abundant adrenal cortical hormone during fetal life. Sex hormones like estrogen, progesterone, and testosterone round out our steroid class here. Oh, and a quick note. Don't forget that vitamins A and D structurally function as hormones too, with vitamin D being central to bone mineral metabolism. Moving on to Section 4, Group 1 Intracellular Signaling, the lipophilic pathway. We're transitioning now from structural taxonomy into the exact mechanisms of action. Hormones are functionally divided into Group 1 and Group 2. Group 1 hormones are highly lipophilic. Because they hate water, they absolutely require carrier or transport proteins in the blood. They have a long half-life, lack second messengers, and their actions are mediated entirely via the receptor hormone, or Rh, complex. Now, what's really interesting here is how strictly defined the boundaries are between their two intracellular receptors. Type 1 receptors are localized in the cytoplasm, they form homodimers, and they are exclusively utilized by steroid hormones like glucocorticoids, mineralocorticoids, and sex hormones. Type 2 receptors, however, are totally different. They are located directly in the nucleus, they form heterodimers, and they're utilized by T3, T4, vitamin A, and vitamin D. But remember, despite these structural differences, both pathways ultimately converge to drive gene transcription. Let's step through the type 1 steroid homodimer signaling cascade. This is a critical 6-stage biological pathway, and we can't skip a beat here. 1. The inactive steroid receptor sits in the cytoplasm, tightly bound to HSP90 heat shock proteins, which essentially act as molecular chaperones keeping it dormant. 2. The lipophilic steroid hormone enters the cytoplasm, binds to the receptor, and forces the immediate dissociation of HSP90. 3. This triggers a profound structural configuration change. 4. The formal receptor hormone, or Rh, complex is now generated. 5. This activated Rh complex physically migrates from the cytoplasm directly into the nucleus. 6. The complex hunts down and binds specifically to GREs, glucocorticoid-responsive elements, on the DNA to initiate gene transcription. Make sure you don't omit a single step there. By contrast, let's look at the type 2 nuclear heterodimer mechanics using calcitriol, which is 1,2,5-dihydroxy-D3, the active form of vitamin D. Calcitriol totally bypasses the cytoplasm. It enters the nucleus directly and binds the vitamin D receptor, or VDR. But can the VDR act alone? No. It has to pair with the retinoic acid receptor, the RXR, forming a heterodimer. Together, this complex binds vitamin D responsive elements, or VDREs, to drive transcription. This heterodimer activation demands co-regulator binding, or the dissociation of co-repressors to silence or activate genes. And thyroid signaling perfectly mirrors this. The thyroid receptor pairs with RXR, and together they bind thyroid responsive elements. Section 5. PPARs and Orphan receptors. Clinical pharmacology and gene silencing. This is exactly where basic science meets the prescription pad. Let's look at the formal definitions you need to master regarding synthetic pharmacology and ligand-independent activation. PPR stands for peroxisome proliferator activator receptor. Much like the VDR, PPRs are heterodimer receptors, but clinically they are activated by highly targeted synthetic pharmacology. Orphan receptors, on the other hand, are fascinating. They act as constitutive activators or repressors. What does that mean? It means they require absolutely no ligand binding whatsoever to execute their function. So the crucial point is knowing exactly which drugs hit which receptors. PPR alpha agonists are your fibrates, like clobefibrate. We use these specifically to treat hypertriglyceridemia, primarily to prevent pancreatitis. And the exact tested threshold for treatment you need to know is triglycerides greater than 440. PPR gamma is targeted by pioglazone. It fights insulin resistance, much like metformin, but watch out. It's heavily associated with severe adverse effects like salt and water retention, bone fractures, and bladder cancer risk. Hardly the side effect profile you want, right? Enter seroglitazar, a dual PPR alpha and gamma agonist. This is the modern drug of choice for diabetic dyslipidemia and is highly favored over pioglitazone as a first-line therapy for NAFLD and NASH. On the orphan receptor side, memorize 3 pathologies. SF1, steroidogenic factor 1, and DAX1 are constitutively required for gonadotrophin and adrenal cortical development. Finally, any genetic defect in the orphan receptor HNF4 alpha directly causes MODY type 1, which stands for maturity onset diabetes of the young. Finally, Section 6, Group 2 membrane transduction, hydrophilic cascades and second messengers. We're pivoting now to our Group 2 hormones. These are hydrophilic, meaning they're water-loving, so they absolutely cannot cross the lipid bilayer. They have a short half-life, require 0 carrier proteins in the blood, and must bind to extracellular cell membrane receptors. There are 4 major classes here: G-protein-coupled receptors, tyrosine kinase receptors, JAK or cytokine receptors, and serine threonine kinase receptors. Let's meticulously walk through the most famous of these, the GPCR pathway. In its inactive state, the GPCR has an alpha, beta, and gamma subunit, with the alpha subunit bound to GDP. Step 1. The hydrophilic hormone binds the transmembrane receptor. Step 2. The receptor undergoes a rapid structural configuration change. Step 3. That inactive GDP is swapped and converts into active GTP on the alpha subunit. Step 4. The newly formed alpha GTP complex breaks away, dissociating from the beta and gamma subunits. Step 5. The alpha DTP binds the effector enzyme to generate a massive second messenger signal. Note that group 2 effects occur mainly through transport channels, protein translocation, and protein modification, not the immediate gene expression we saw with group 1. So, the GPCR pathway generates 3 main second messenger systems. First up is cyclic AMP via GS or GI proteins. Here, adenylyl cyclase forms CAMP, which activates protein kinase A, driving downstream protein phosphorylation. Second is the IP3 DAG system via G2 proteins. Phospholipase C cleaves PIP2 into DAG, which activates protein kinase C, and IP3, which triggers the calcium calmodulin pathway. This pathway is famous for vasoconstrictors like endothelin and angiotensin II, but do not forget neurotensin. It is unapologetically the most potent vasoconstrictor in the entire human body. Lastly, we have cyclic GMP via GT or transducin. Unlike the intense vasoconstriction of IP3 DAG, cGMP is famously linked to vasodilation, acting through nitric oxide and atrial natriuretic factor. Let's move to synthesize this into our ultimate high-yield takeaway and see how this all builds together. You must be able to map these exact hormones to their specific GPCR second messenger. From the hypothalamus, CRH uses cyclic AMP, while TRH and GnRH use IP3 DAG. From the anterior pituitary, TSH, ACTH, FSH, and LH all use the cyclic AMP pathway. But listen carefully to this exception. Growth hormone and prolactin do not use GPCRs at all. They uniquely operate via the JAK-STAT cytokine receptor pathway. You absolutely have to memorize that matrix. We have covered a staggering amount of academic ground today, from amino acid counts to exact intracellular signaling cascades. So let me leave you with a rigorous question to integrate what we've learned. If a single genetic mutation completely disrupted a receptor hormone complex's ability to physically migrate through a cell, would the resulting clinical pathology resemble a group 1 intracellular deficiency or a group 2 membrane transduction deficiency? Think about the precise mechanisms of type 1 cytoplasmic homodimers versus GPCR extracellular pathways. Revisit your notes, solidify these structures, and I'll catch you on the next explainer.